Good evening, my fellow scientists. It is Wednesday, December 13th, 2017, and I want to talk about a new and possible cure for Huntington's disease. Huntington's disease is a terrible genetic condition where a mutation results in a problematic protein. That protein is expressed in the brain, it breaks apart, and eventually accumulates in sufficient quantities to kill a person who has it. So it's a really just terrible disease. There's no known cure. The best that has been accomplished is to have a good test for the disease so that people can know whether they're going to have it and whether it's going to be severe. But beyond that, it's just make your peace and plan for the inevitable. As of yesterday, it may not be inevitable. The Ionis HTTRX drug was released publicly. The trial results were released publicly, and the trial results are actually quite promising. This is still a small-scale trial, so I don't want to read too much into it, but the notion is that a small piece of single-stranded nucleic acid, so like half of DNA, is directly injected into a patient's nervous system, into their cerebrospinal fluid, and that piece of nucleic acid migrates into cells, where it interacts with the messenger RNA that is coding for that problematic protein and suppresses the production of that protein. This is the first example that I had read about a successful trial of a antisense therapy. It turns out there have been a couple more over the last decade or so that have not made quite the same news, probably because there are other options for many diseases, but in this case, there's no other option. And so going after this protein and suppressing its production seems to be kind of the only way to approach the problem. The particular challenge here, of course, was that to make this work, you need to get the drug across the blood-brain barrier, and getting a huge DNA-like molecule across the blood-brain barrier is almost impossible. To get around this, they injected it directly into the cerebrospinal fluid. That's less than ideal. Getting a drug through the blood-brain barrier would be preferred if there was some way to shuttle it. There's not. That's a huge active area of research. If it can't be circumvented, then you need to inject it into the brain. Minimizing the number of times that has to be done would be ideal if you could have a controlled release scenario where you inject some substance that then slowly releases this type of drug into the central nervous system. That would obviously be an advantage. So there's a huge amount of work to be done here, even if this drug proves to be really successful. So I'm excited about that because that means that there's going to be more interest in the nucleic acid field, nucleic acid therapeutics. That there's a niche for those is exciting to me because I work with nucleic acids, synthetic nucleic acids especially, and I am excited to see this come out. So if you like hearing news about science and chemistry and biochemistry and nucleic acids, tune in Monday through Friday. We talk about all that and occasionally some results from the Allen Lab.